The cell cycle is a process of cell growth, specialization, and replication during which an ordered sequence of events takes a cell from its origin to division into daughter cells.
There are two phases of the cell cycle for eukaryotic cells. They are interphase and M-phase. Interphase is the longest stage of the cell cycle and it is split up into 3 phases. During G1, cell growth occurs and the cell decides whether or not it is going to divide. In S phase, DNA is replicated. A cell spends almost half of its time in this stage. G2 is the phase during which organelles are copied and the cell prepares to divide.
M phase consists of mitosis and cytokinesis. Mitosis is the division of the nucleus and its genetic material. Mitosis is important because it allows cells to divide and pass their genetic material on to identical daughter cells. Cells also divide to repair damaged cells. Mitosis results in genetically identical daughter cells with the same number of chromosomes as the parent. Mitosis is composed of prophase, metaphase, anaphase, and telophase.
During prophase, chromatin condenses into chromosomes, the nucleolus disappears, and the nuclear envelope breaks down. Chromosomes move to the middle of the cell and the mitotic spindles begin to appear. In metaphase, the chromosomes line up on the metaphase plate and mitotic spindle form and attach to the centromeres of the sister chromatids. In anaphase, the centromeres of sister chromatids separate and the sister chromatids become chromosomes. Spindle microtubules pull the chromosomes to opposite poles of the cell. Telophase occurs when chromosomes reach opposite poles of the cell. 2 nuclei form, which will make up 2 new daughter cells. Cytokinesis is a division of the cytoplasm following telophase. In animals, microfilaments form a cleavage furrow and the cell pinches into 2. Rather than pinching, plants form a cell plate made of membrane vesicles that split the cell into 2.
Meiosis is the cell division in reproductive cells. It divides chromosomes of diploid germ cells in order to create 4 haploid gametes. Meiosis is important in creating genetic diversity among organisms. Like mitosis, meiosis is preceded by interphase. There are 2 stages of meiosis. The purpose of meiosis 1 is to separate homologous chromosomes, while meiosis 2 separates sister chromatids. During prophase 1, replicated chromosomes coil and condense. Sister chromatids are joined together at their centromeres by cohesions. The nuclear envelope begins to break down. Synapsis occurs, which is the binding of homologous chromosomes. 2 sister chromatids bind to each other in synapsis, which forms a set of paired sister chromatids composed of 4 chromatids called a tetrad. Crossing over also occurs during synapsis. Crossing over is the exchange of genetic material between homologous chromosomes. In metaphase 1, tetrads line up randomly along the equator of the cell parallel to each other. This random alignment is known as independent assortment. Spindle are visible, and microtubules connect to the kinetochore of each homologous chromosome. In anaphase 1, kinetochore microtubules pull chromosomes toward opposite poles. Pairs of homologous chromosomes split up. Telophase 1 begins when the separated homologous chromosomes reach opposite poles. Chromosomes uncoil and nuclear envelope begins to form. Cytokinesis creates a cleavage furrow which separates the cell into 2 daughter cells. During prophase 2, spindle apparatus forms and the nuclear envelope disappear. In metaphase 2, chromosomes composed of 2 sister chromatids line up along the metaphase plate. Spindle fibers attach to the kinetochores of sister chromatids. The centromeres of sister chromatids separate in anaphase 2, and sister chromatids are moved to opposite poles of the cell by kinetochore bound to microtubules. Sister chromatids become chromosomes. In telophase 2, the spindle apparatus breaks down and the nuclear envelope forms once the chromosomes reach the poles. Chromosomes also uncoil. Finally, cytokinesis produces 4 haploid daughter cells with unique sets of genetic information.
A difference between mitosis and meiosis is their functions. The purpose of mitosis is to produce 2 genetically identical cells, while the goal of meiosis is to produce 4 genetically unique haploid gametes. In addition to this, daughter cells produced by mitosis contain the same number of chromosomes as the parent cells, while daughter cells produced by meiosis contain half of the number of chromosomes as their parent cells.